Well, they aren't mine. They're Elizabeth's.
No, I didn't see them yet.
I didn't see them. Right? Did you send them to me?
Okay. I didn't send them to you, Bradley, but I could. But this is just.
When did you send them?
A week ago or so.
Oh, a week ago. Well,
Yeah.
Well, let's see. Do a search on Susan. Nope, nothing comes up. Better send them again.
Oh no. Okay.
They've been censored. No sex between X-Moms allowed on screen.
Yeah, something. No, you catch COVID from the pictures.
Might turn on all the X-Moms in my house.
Well, one was a pinhead.
Oh, good. Okay.
Yes.
Hello, everyone.
Hi.
Hello.
Yeah.
How
Hi.
are you?
Yeah, I'm fine. How are you all?
Oh, pretty good.
I'm glad you're good. We have some issues here. All my children have decided to catch COVID.
Oh.
Why so?
They live near Steinbach, or they work with people from Steinbach, and Steinbach has, they said, 40% infection rate. Yeah, that's a hot spot. Never mind, they're all younger,
Yeah.
and they're staying away from me, so that's fine.
Yeah.
Hello, Mayukh.
So,
Okay.
I think he's there.
Yeah, Susan, please resend those. For some reason, I can't. Oh, maybe you sent them to Gmail, which that would be fine.
Oh, I sent them to your Gmail. I send them to Natalie's Gmail too.
She doesn't use Gmail either. Nope. We're boycotting.
Okay. All right, I'll send them to a more appropriate address.
Okay.
Okay. I don't know what her email address is. Show them to her when you get them.
Oh, okay, sure.
Okay. Yeah, I can do that.
Okay.
Ah.
You want to see them, Bradley?
Oh, yeah, I would. Yeah, you can send them.
Yeah, this is what can go wrong in crossbred axolotl.
Oh, okay. So, welcome to the meeting. I guess we have a couple things today. I think Mayukh wanted to get back in touch about the JOS paper. Then Krishna, I think, did you have something to share? Or?
no, not
Okay.
today.
All right, then I have a couple papers I pulled up, and I think that's it. Maybe some other things as well. Not really sure what I have in here. Oh, Susan sent those optical tomography papers from last week.
Yeah, I saw them.
Yeah.
Yeah. I'm
Did you
working on
want to?
them.
Did you want to talk about those a little bit, Susan?
Uh,
Oh, you're muted.
I have a small, just have a very small group of slides about it if you want to see them.
Okay, that sounds good. Yeah, let's see.
Would you like to see them?
Yeah.
yeah.
All right, so I have to share the screen. How do you do that?
If you go down to the bottom, present now at the lower right-hand corner.
Oh, there it is. Okay. Okay. It doesn't want to.
Huh. It should say, well,
I'll turn off my video,
okay. Maybe that's it.
so you won't be able to see me. But hi. There, I'll just turn it off. Just like this. There we go. I'll try again. Maybe I'll just. There. No, it doesn't want to. Okay, we'll try another tactic.
Okay.
Okay, it doesn't want to share.
Hmm.
Sorry. I have I have, it's only 8 slides, so.
Oh, okay. Well, do you
Can
want?
you send it? Bradley, can
I
you
can-
put them up if she sends it to you?
Yeah. Well, can you send them to? Well, share. Can you share a link? Are they on Gmail or are they on like a something where I can share them or?
Yeah, I could send them to you.
Okay.
Hmm.
Well,
Where is
anyways, why don't you just something you can talk about, or is it something that like you need to present?
It's just that I'm kind of working in this area. You get optical coherence tomography, and you can either take a whole bunch of pictures all at once with like a white light, a very bright white light, and get all of the different frequencies, including infrared, and then combine the pictures to get a good look at, say, skin. Or you can use a swept source, which sweeps through the frequencies and get a combined image that way. And then we're trying to add optical coherence elastography by moving the tissue and watching how it responds. And that's sort of like doctors used to massage tissue to see what was up with it. Because if you've got cancerous tissue, it's has different mechanical properties than the other tissues. And anyways, so we're trying to make some optical coherence elastography images as a part of my PhD.
Okay.
Anyway, so
Susan, I have a question.
yeah.
The elasticity of a volume of tissue can, of course, be anisotropic and represented as a tensor.
Yes.
Now, does your math allow reconstruction of tensors?
Yes.
Oh, it
Yeah,
does.
that's where Sharif comes in, because he's very good with tensors.
Oh, good. Okay.
Yeah, that's his lab does tensor math all the time for the optical coherence tomography that he does.
Oh, okay.
Yeah, so I just sent
Okay.
the slides to you, Bradley.
Okay.
Okay, can you do me a favor? Does he have some papers on how to use tensors for optical elastography? I guess.
We're just getting into that. I have some. I'll send you Wasinga, his PhD thesis.
Okay, good. Thank you.
He's the best paper. So I don't know how long it takes to send slides, but anyway, they're I sent them to you, Bradley.
Okay.
I responded to DevoWorm number 44.
Okay.
Anyway, they're only 8 slides. It's just some sample images showing that what's possible, and the idea is that you don't need dye and you don't need to cut the tissue. You can do it live, and they're developing a device that will do cancer margins while in the operating room with this. That's the Kennedy Wasinga group. So,
The margins have a different texture from the tumor and the surrounding tissue?
yes.
Oh, they do? Oh, neat. Okay. Because classically, the physician just guessed.
Yes, I know.
Yeah, and then the margin, do you know what a margin is, Bradley?
I'm not sure. Oh, you mean the margin?
Okay,
of
okay. A margin is based on the following assumption. Let's take a skin cancer.
Okay.
Okay, the skin cancer cells are assumed to be able to move, and the margin is a guess as to how far they can move. So you not only cut out the tumor, you cut out the surrounding tissue, and that's called the margin.
Okay.
Okay, and then they send the margin to a pathologist to see if there are any cells near the edge of the margin. If there are, then they got to cut more. The margin becomes bigger.
Oh yeah.
Okay.
Okay.
So it'd be very nice to have a direct measurement of what the margin actually is.
Right. Right.
Okay. Up till now, it's been totally guesswork.
Right. I'm still not getting the slides. So why don't we, yeah, that's, so I have the papers here in a folder.
Okay, well, just I had just chose a couple of images out of the papers just to show that it works. Like they were nice images, so you can share the slides if you want, or just read the papers. That they're the one on optical coherence elastography is a little bit more involved, but that's okay.
Yeah.
Yeah.
Yeah, so here are the papers here. So this is optical coherence elastography for tissue characterization. So this is a review paper. And this is, let's see, this is the first figure here, which is, I guess this is the position of OCE among other elastography techniques. So if I zoom into this, you can see that I like these kind of graphs because they show kind of like the space of I've seen these for neuroimaging too, where they have like the spatial resolution, which is on the order of, 1 millimeter and 10 to the negative 1 millimeter, 10 to the second millimeter, which is, they're different distance scales centered on a millimeter. So, and then you have the field of view.
Nice.
Yeah.
To have this, because then you can see the whole thing, and then you can see close up, like you can do scaling. So that's why that's nice, and it also does fairly fine imaging.
Right. So OCE is at this red. Oh, there's Jesse.
What were the abbreviations on that?
Okay, so OCE is optical. Let's see.
Oh, I see. Those are the different methods.
Yeah, different methods. AFM is, I think, atomic force microscopy. OCE is the optical elastography. I don't know what some of these others are. They don't really have it in the...
I have them in the slide.
Oh, okay.
They're around. They're there in the paper someplace.
Yeah.
They're just. I thought they were just below it, but anyway, ultrasound, MRI, atomic force microscopy. Yeah.
Yeah. So this is the field of view spatial resolution. So this is like how, when you, like, it's like microscopy, where if you have a higher resolution, you can get to a smaller spatial resolution. And then that's connected to the field of view, which is if you have a single image, what distance does it cover across the image? So you see atomic force microscopy is way down here, and then OCE is up here.
Yeah, one problem with this is that it doesn't take into account Google pixel imaging.
What is that?
Oh, that's where you montage a bunch of pictures together at high resolution.
Oh, okay.
So you get a much larger field of view.
Oh, okay. Yeah. So
OCE, you can do that with optical coherence elastography. You can stitch images.
I mean, you can do it with any method you can get a picture from.
Yeah.
You just need to have your camera, whatever it is, have a much larger traverse. But it's widely used. I just saw a webinar on where somebody took a whole microscope slide, 1 by 3 inches, and they scanned it at very high resolution, the whole slide at 60 depths. So they got depth of focus also out of it, and then they montaged the whole thing. So you can look at it at any resolution you want. And it's called gigapixel imaging.
Oh, wow. Yeah, I have not heard of that, but I'll look into it.
Oh, okay. Yeah, it's a fairly large field now.
Oh, okay. Wow.
Yeah, it's used for many different things. If you take any microscope, if you can get precise enough motion of the specimen, you can then move from one part of the specimen to another and then montage them. So the key is either to move the specimen very accurately or to have very accurate montaging software.
Yeah. I'm just taking some notes.
Yeah, it's been used, for instance, to take a picture of a whole crowd of people where you can focus down on one face and see the face in high detail.
Oh, wow.
Okay,
Yeah.
so it's used in many different fields.
Yeah. So this is the paper here, the first paper. They go into a lot of the detail. Oh, go ahead.
Yeah, so I'm just saying the field of view is not really a limitation if you go to gigapixel methods.
Yeah, yeah.
It can take a long time because for some machines it takes a long time to acquire each image, which gets montaged.
Right.
So it depends on it does depend on the method as to whether it's practical or not.
Yeah.
Do you have a paper on that?
Gigapixels. Let's see if I can find some.
Okay.
I'll look.
All right.
Okay, go ahead.
All right. Yeah. Yeah. So this is. Let's see if we can find another image here, because they just kind of go through the technical details of OCE, and then some of the math and displacement, amplitude, or strain. So they go through sort of Young's modulus and how they calculate that. Then they go through some more math, and this is on the viscoelastic models. So they talk about like the mathematically modeled the tissue. What else? I'm trying to find another image here, but I don't. I think that's the only image they have in this paper.
No, it's at the end.
Oh, there's something at the end.
Very end of it, yeah.
Okay, it's probably
They put all the images at the end.
Oh, okay.
There it is. This is the one I wanted to show. Whoops.
Which one, figure 8 or figure?
Well, this one too. Actually, I should have included this one because there's OCT and then OCE and then this histology image, and so.
Yeah. So this is a comparison of OCE, and then this is a comparison between that and. So this is a microelastogram here. And then this is the histology, which is like where they take the tissue and they stain it for different markers. So yeah.
Yeah, and you can see that the OCT and the elastogram combined will give you as much information as the histology image. So that means you can do it without dye and living tissue. That's what you're looking at.
Yeah. And then this figure 9 is, this is another one you wanted to show, or?
Oops, at the top, this is just the signal you get back down below. And the top, whoops, there it is.
Yeah.
You can see the difference between the adipose tissue and the tumor. And then the histology image is in pink. So
Okay. Oh, yeah. So this is the tumor and adipose tissue. So this is the
So you-
difference. Yeah.
yeah, you can see the difference right before your eyes. And like I said, in living tissue and without dye. So that's a huge advantage.
Yeah. And so this is the margin in this area here in the middle, or?
Yeah, it's just wherever you've got tumor-like image. So the tumor's there, and I can't show you, but yeah, you can see the difference between the tumor and the adipose tissue, so you would know where to cut, basically.
Yeah. Yeah. I see. Okay. And then were there any other images you wanted to show, or?
No, that's okay. This
Okay.
whole, the end of the paper here, you can see a whole bunch.
Yeah, there are a lot of nice images in the paper, like at the end of the paper.
Yeah, it's toward the end.
So this is a quantitative elastogram. So this is kind of like where they actually quantify so they get at the pixel level, I guess. So they're actually quantifying the strain, I believe, or yeah. So that's that paper. Then the other paper is Multi-Spectral Near-Infrared Absorption Imaging for Histology of Skin Cancer. So this is a similar thing where they use near-infrared images. So this is... are there any images from this paper you want to show, or?
Well, they're just a setup. It's multispectral optical coherence tomography is down the way a bit. It's how they do this. That's the one method of doing it is if you keep on going. There's multi-spectral images. There's the setup.
Okay.
And you have a lamp that produces multiple wavelengths, and then you have a camera that takes the image at multiple wavelengths, and then you can combine the images to get a good histology image of the tissue.
Yeah.
And it's all at this 1100 nanometers, which is kind of a water window. They call it a water window, where you get the most penetration into a tissue.
Okay. Yeah.
Naturally occurring water window. Okay, I have several interesting things happening with me, including a dog barking and stuff. Anyways, then there's some images that of this skin that there they go. There's all of the different frequencies, and you can see at 1000, 1100, and 1200 that you've got pretty clear images. 1300 as well, and they say that it's not. They didn't set it up correctly for the 900.
Yeah.
It's not optimized for 900 nanometers, but you could probably do better.
Yeah.
And if you keep on going down, and you can see these three histology images compared to optical coherence tomography and optical coherence elastography, I believe. Anyway, you can see detail.
Yeah.
Quite decent detail using that method. Pardon?
Yeah.
Yes?
Susan, all of these pictures could be made much sharper by running through histogram equalization. Okay?
I'm sure they've tried that.
I doubt it. It doesn't look like it at all. They look pretty washed out.
Okay.
Yeah, you find it in Image J.
Yeah, you can do it in MATLAB too. I've done exercises in histogram equalization. Yeah, it's okay.
Okay. Yeah, but they, you know, these ones that are very light gray, they're hard to compare for detail without doing.
Well, if you what they normally do in MATLAB is do that pseudo color for the different levels of gray, and then you can really see it.
Yes, except pseudo color can give you artifacts in imaging.
I suppose if you were a doctor, you'd want to see the real thing and then mess with it yourself to see if you can see more detail.
Okay. Okay, good. You might give it a crack with some of these pictures, see what they look like.
Well, I'll try to get some of my own and see what works best with them. Yeah, MATLAB has a lot of imaging tools in it,
Okay.
including wavelet tools.
Yeah.
Yeah. Anyways, yeah, ImageJ is good too, and it's free.
Yeah.
MATLAB is not, although the university here decided they were going to buy it for their students. Hooray! Finally.
So these are near-infrared variance images of unstained samples. Is there any other pictures you wanted to show?
No, that was all. That was the best three, the combined three. And this is skin, so it's a little easier than adipose tissue. I'm going to turn off my, yeah.
Okay. All right. Well, yeah, thanks, Susan. That was good. So I think you got an idea. A lot, you know, there's some of you here probably don't, you know, maybe a little bit above your head in terms of the technique and all that. But so we're looking at different ways to image tissue here. We have the regular microscopy that we usually feature in the meetings, but that's usually on something that's fixed or, you know, a culture of cells or of something else, maybe worms. So we're taking, you know, we can take images like that, but then we can also do histology, which is where we take images of tissues. Sometimes they fix the tissue with a chemical solution, and they slice it up and they put it on a slide, or they'll do it live. And this is kind of what we're talking about, is doing this sort of imaging live, where you put an imaging device on like someone's skin, and you're actually looking at the skin live, and you want to see detail underneath. So like they're looking for cancer, tumors, or they're looking for other things in the skin, you can see them. And Dick has done a lot of work on that. So Susan is actually looking into some of these methods. So this is all really interesting. I mean, you know, if we want to, I sent out these two papers last week. There are other papers, of course, that, you know, I think Dick probably has bibliographies on this. If he's, you know, willing to share them, if you're interested, you can get in touch with him. And,
sure.
Yeah. So, next, I guess the next thing we'll move to is Mayukh. He got back in touch about the the Journal of Open Source Software paper. Okay, there's Dick Gordon's email here if you want to contact him. So let's see. The paper is here. Let's see. Let me share my screen, because we've got. I was presenting. Okay. Looks like we have Assaf here. So he's been around OpenWorm a bit, and I've talked to him before. How are you? Oh, you're muted.
Well,
yeah, I'm okay. Nothing new by me. I'm listening in. Interesting stuff.
Yeah. Well, welcome. Yeah.
Thank you.
Let's see. I'm gonna share my screen again here. So we'll talk about this. This is the paper that kind of came out of last year's Google Summer of Code. So we created this DevoLearn framework, but we also created the DevoLearn software package, and that was Mayukh's project. So this is the repository. It's on DevoLearn on GitHub. So GitHub, we have a DevoLearn organization, as we know. For some of the new people, maybe you've not seen this, but we have different things going on here. So we have education, theory building, some data science demos. Then we have DevoLearn, which is the standalone software, and then C. Elegans DevoLearn, which is actually Ujwal's project from last year, which is taking a lot of the resources that we've had up to this point and putting them into a library of machine learning tools. So we have machine learning resources, deep learning resources here, but we also have some other resources in terms of education, in terms of just general data science, like how do I analyze data once I get it get it processed by these algorithms. And then this I'm thinking here of putting together a guide for model organisms, which is what we originally had in DevoZoo, but more so newcomers could come in and learn about an organism like zebrafish, just basic information. Because, you have people who might be coming here who are computer scientists or, they've not heard of some of these organisms. And so it'd be nice to give them some background on some of them. Nothing nothing too comprehensive, but just enough so that they know, what they're looking at if they have a data set that they're using. So this is the JOS paper. So this is all sort of organized. Actually, this repository has some images and some video. Yeah, so this is where the paper resides. This is the manuscript here. And I don't know if I have this. I think I made a mistake on the formatting here, but this is the title. So pre-trained deep learning models that enable computational developmental biology research and education. It might be a little long, but I'll think about that more. So it's Mayukh, myself, and Ujwal, and this is our affiliations. This is the format that they want. So the Journal of Open Source Software, it's run on GitHub. It's a peer-reviewed journal, but they run everything on GitHub. So you're supposed to submit a markdown file in a certain format, and we need to finalize that format. I think this is not quite right, but I don't know what it is. I think Mayukh, maybe he had it in a certain way, and
change. I've been reading this today, so there will be some changes that are to be made. So I'll make them soon.
Okay.
Within today.
All right, that's good. Yeah, you please make whatever changes you need to make to if they're wrong.
I'll also have to discuss with Ujwal maybe.
Yeah.
Because let's see, there are some changes which I'll have to ask him about.
Okay.
Yeah.
Yeah, I'll get in touch with them. You can get in touch with them. So, but I want to look it over.
On Slack. That will be better, I guess.
Yeah. So, so here we are. So this is so the summary. We have some it's a summary of what was done. This is for the software package, and then the technical details. So I'm sure that I put this together this weekend, just kind of walking through. This was taken kind of from the Summer of Code report. So Mayukh might find some problems with it. I mean, if you want to add to it and add detail or make it clear.
No, there are no problems actually, but I'll just have to make some minor changes.
Okay.
Some of the sources are not right, so I'll just change them.
Okay, that sounds good.
Even I forgot to save some of the sources because I was actually looking for the sources. I forgot the data sources, and then I had to find again.
Oh yeah.
Yeah.
Yeah. I mean, just a point on this. We want to be. It's important to be able to cite where we got the data from. Some of these open source tools that we cite, or some of these open source repositories, you know, if they want to get funding, they have to have citations of them by other people. So if, you know, we can cite them and then they have those in support of like their further development, that's a positive thing. So we want to make sure to cite them as much as possible. So that's in this part. So we mentioned like the Epoch data set, the worm image database, and then we've given a citation, and then that will help them out. Then I redrew some of these figures. So this is the figure, sort of like a schematic for the software. So this is where you have the GitHub source, what's on GitHub, and this is the user environment. This is for DevoLearn. I should probably change it to the version number 2. And then this kind of describes how it works. It's very data science friendly, compatible with NumPy and Pandas. And then this is an image of the umbrella. So then we could start talking about transitioning from the DevoLearn program to the DevoLearn framework, which is this framework where you have DevoLearn, the program here, and then this larger framework of things. And then that's where like Ujwal's project is here, and then this is some of the existing infrastructure. And then I kind of mentioned that because, you know, it's like people will be able to go to the GitHub organization and contribute, and it'll drive everything forward. And then we have some acknowledgments here. And then the references. So the references, I think that's good. But Ujwal, or Mayukh or Ujwal, if you want to add in any references, you can. Just be mindful of this numbering scheme, because, you know, if you put in a strange number, it throws everything off. So if you put in something, try to find where it belongs in the numbering scheme and then change it. And then a word on the references. I think this is the way they want the paper is submitted. So they want it with the references in like this. We also have the references in this bibtex format. So I have everything in the bibtex format. So if we add in references, we can add them in like this. And this actually makes it compatible with LaTeX. So we can, you know, add in. We have that in the repository. So the idea is that we'll submit this soon to the Journal of Open Source Software. They'll review it, and then it'll become like a published thing. They'll give it, I think they assign it to DOI, and then, you know, it's a small publication. It shouldn't be any longer, I think, than 2 or 3 pages. And then, you know, then we'll have this, yeah, I think it's pretty prestigious, but it's the idea, I think, is it'll get exposure to a larger audience of people. So there'll be a lot of open source software people who will see it and maybe, you know, promote it or maybe contribute to it. We'll see. So any questions on that?
Yeah, put one in the chat.
Okay, let's see. Do any databases for references recognize GitHub repositories? I'm not sure what that means.
Well, for instance, Web of Science has a number of databases that they recognize,
okay.
and they index those.
Oh, okay. I'm not sure how they deal with them. We know that like GitHub, a lot of people, when they want to publish something that they've done on GitHub, like a release, they'll often use like a third party like Zenodo, and it generates what they call a DOI. And some of the databases will pick up like those sorts of things. But I'm not sure that the Web of Science picks up like those sorts of citations. Now, the Journal of Open Source Science, I don't know if they're indexed. They may be indexed. But, you know, there are databases that look for things like open data sets and things like that. So it's a kind of a gray area because it's always evolving and changing.
Yeah.
Yeah. So, Mayukh, you wanted to present on something called torch streams. Are you still going to do that, or?
No, actually, like I won't like it could be an impromptu presentation, but like I didn't think of anything. Like I could show what it's about and how
Yeah.
it's kind of related to this field.
Yeah, go ahead.
Yeah. Then I'll just share my screen real quick, or you could just show the
yeah,
library, and I could just say
I'll
just a
stop
small
presenting.
thing.
Yeah, there you go.
Okay, I'll share my screen then. Okay, so is my screen visible?
Yeah, there we go.
Okay, so I'll quickly go to the repository. So basically, like what happens in developmental biology is that like we see patterns which emerge from basically like seemingly random things, right? So we can see like similar things which happen inside, like which can be done using neural networks. So basically, like for example, I'll just show an image quickly. For example, we have this noise. Like this is basically random noise, right? But if we feed this image to a neural network, it starts hallucinating certain kinds of patterns. Like different layers within the model hallucinate different kinds of patterns. And if we optimize this noise to further optimize for those pattern hallucinations, we basically get a lot of very like fascinating patterns within deep learning models, and they vary from like model to model. So I basically made this tool which can be like used to visualize certain patterns. So I'll just show this one. So like these patterns emerge from that like these emerge from noise basically, and they are like being visualized by the neural network. So I could go in depth in some presentation soon, like within like maybe next 2 weeks maybe. But for now, all that you have to know is that this library can be used to see how neural networks see the world. So yeah, basically that would be my 1-minute presentation about this thing.
Wow, so that's pretty good. So that's generated all from like noise or like pattern.
It's basically like for example, like when you like I'll give a quick real life example. Sometimes when you look at clouds, you sometimes see trees or even some animals on clouds, right? That is when you are basically hallucinating, quote unquote. So even neural networks, when we feed them those kinds of random images, they have certain activations of animals or even like castles or even palaces and things like that. So even like we can optimize that input image such that those hallucinations they get enhanced further. So we can see what the neural network was hallucinating or seeing in that random noise. It could be noise or it could be even a natural image. And if we feed like more natural images, we get more higher level features like sometimes dogs or even castles. I could show another example which looked like castles. Wait, I'll show it.
Okay.
Let me share my screen.
All right.
Okay, so is my screen visible again?
Yeah.
Yeah, let me just quickly navigate to the notebook. And even like this library has a starter notebook on Colab, so you guys could try it out. So wait, let me just. Okay, so as you can see, like, wait. So on the top, like we can see some random noise that I generated, and then I like basically optimize this image to like visualize some activations within the neural network, and I got things like these. Like these patterns, they don't look like anything, but they are like chains or like ropes or something like that. And then again, I optimized another layer, and we can see something like grass or like I don't know what this is, but this is some kind of a pattern that's emerging after like 500 600 iterations of optimization. Like I am back propagating on the image instead of the deep learning model. That is what is happening inside.
Okay.
So like if I want to summarize it in one statement, it's like if it looks like a cat, make the image look more like a cat. That is basically a one-line summary of what's happening inside. So then then what I did was that I like instead of optimizing these two channels individually, I optimized them together simultaneously. And after doing that, I got something like this, which is a mix of both. And we can even see eyes, things like eyes and stuff. So it's kind of fascinating for now. I plan to explore further, and I want to see how this kind of works out, or maybe I think this has some analogies with cellular automata and like automata and all that stuff. So yeah, I'm exploring right now.
Yeah.
Wow.
Yeah.
Are there any papers on that?
Yeah, there are papers actually. Like one of them was, I've been reading a lot. I'll drop a few links then. Like there are a few good papers on these things.
Yeah.
Okay, very interesting. It shows how untrustworthy these methods are. Okay, basically they can acknowledge anything we fancy.
Yeah. Well, there's
Okay.
actually, yeah, there are people doing research too on like visual illusions and like neural nets. And I think there's a guy in Japan who does a lot of work on this, where they're looking at. I think a long time ago I did a presentation on what is the name of that thing? It's where you perceive faces and things, and that don't have faces, and so those are things that we can treat as errors, and they are errors, but like it opens up an interesting avenue as to like what the networks are actually doing, because we do that a lot.
You're deep dreams.
Right.
Very interesting.
So, well, that's good. Yeah, if you could share some of those links, Mike, it'd be great.
Yeah, I'm sharing. Like I'm looking for them right now. I have them all saved.
Okay. So yeah, that's good. So
These are not focused on biology. These are more focused on how the neural network works. So it might still be interesting. At least the photos look really good, like which get generated.
yeah, that's good. So does anyone else have any news they wanted to talk about, or like any updates? Krishna, Jesse, Shruti.
I want to ask something to Susan regarding the mechanical index. What that MI thing is in, you say OCE, and am I being audible?
I think she's muted. I don't know. Well, you can ask the question.
Okay, there. Sorry, what was the question?
Yeah. What is MI and in this OCE thing?
What? Sorry, just I'm online. Just a minute. So what was it about the OCE thing?
What?
Yeah, I want to ask what is mechanical index and how does it value, you can say, deflection of its value, how does it affects the thing that we are going to, how does it affects the elasticity that of something like that we are, you can say, calculating. You want me to write the question?
I think I understand. What you do is you take an image of the object you're testing with optical coherence tomography, which is infrared light, and then you perturb it, like you push on it or squeeze it or poke it or send an air puff at it, and then you take some more images and you compare images. So you can watch the response, the mechanical response of the object, to determine both the actual change in the image and the phase portions of the image to see what the elasticity of the object is, the viscoelasticity.
Yeah. Do we have some, you can say, predefined general elasticity? Do we have some normal threshold range, something like that?
What you do is you put a known tissue or known substance in with the object you're testing, so you get a baseline.
Okay.
Yeah,
Do we have a general baseline for all the human beings, or we make that threshold value specific to a single person? or we have, like, for example, if we are exploring the skin cells, then we have a certain value for that, or do we calculate the skin values of a particular person? Because everyone, you can say the elasticity of skin or any, you can say, tissue can vary from person to person regarding age, or you can say.
Usually it's a comparison. Like they haven't. They're still being researched on. They're trying to get what they call qualitative images, where you can compare this image from this person to that image of that person. But like I said, you need to put in need some sort of known substance, and usually it's a gel that has been made similar to the tissue, and you can test that with other systems to know exactly what its stress strain rate is, and then you can use that to compare to the tissue that you're measuring. But often what they're doing right now is just comparing this tissue in this person over here with the tissue next to it. So they're just doing a comparison within the same body. That's what I understand from my reading anyways.
Yeah, let me make a general comment. There is a general problem which has hardly been addressed, and that is the detection of melanoma, which is a dark pigmented tumor. It can start out very small in the skin, in dark-skinned people, where the visual contrast is low. And maybe these optical coherence techniques might solve that, but it's still an unsolved problem.
Yeah, I think the optical coherence tomography, because it's infrared, that 1100 nanometers I was talking about, it goes right through melanin.
And can it see melanin on a dark-skinned person?
It doesn't. It goes through melanin. That's what I've been reading.
There's no contrast of melanin with normal skin?
No.
Oh, that's unfortunate. Okay.
Well, you can't use melanin as a dye for 1100 nanometers. It doesn't work. It doesn't
absorb in that wavelength.
No, it doesn't absorb 1100, 1300 nanometers.
Do you have a spectrum for melanin?
I found one. You want me to find it? I'll go looking. I had one. Yeah.
That's very useful.
Yeah.
I have one more query. Can we do, instead of using that gel, can we, find a normal range for human for specific tissue? Like you can say that taking statistical mean of 100,000 people, and then you can say find some general tendency of what would be the range. And if that range is exceeded, or you can say interceded, so we can say that there are some abnormality.
One would have to do a lot of comparative testing.
Yeah. Yes.
Yeah.
Well, yeah, that sounds good. So I was gonna, if you have to go now, it's fine, but I was gonna finish up today with a few papers, just kind of go over them briefly. And thanks for your comments. I don't know if Jesse or Shruti had anything to add before we move on. Any questions or comments?
No, sir. As such, I don't have anything.
Okay. All right. So I'm gonna, as our obligatory paper cue here, I know we presented Susan's optical tomography papers, but I have a couple other things to look at here. So, okay, here we are. So I have two papers that I found this week. One of them is really interesting. I think if you're interested in like cell-cell signaling and morphogenesis. It's called inferring the spatial code of cell-cell interactions and communication across the whole animal body. And so this is actually something from C. elegans. So this is something that, this is the graphical abstract. You have cells in the phenotype. This is a model of C. elegans. They just have sort of the cells, laid out here. They don't have the, the usual external part of the worm that we see. But then you have these cells and you're looking at intercellular distances. And then they're looking at those things in terms of transcriptomics within cells, and then they're trying to infer cell-cell interactions. So the abstract is cell-cell interactions are crucial for multicellular organisms as they shape cellular function and ultimately organismal phenotype. However, the spatial code embedded in the molecular interactions that drive and sustain spatial organization, and in the organization that in turn drives the intracellular interactions across the living animal, remains to be elucidated. So here they're looking at the expression of ligand receptor pairs obtained from a whole body single cell transcriptome of C. elegans, and this is in the larvae. So this is after the egg hatches, but before it becomes an adult. So there are a whole bunch of larval stages, as we know, and they look kind of like mini adults, but there's some morphogenesis still going on. Especially, there is some plasticity as well, so depending on the environment, there's some changes that happen in the phenotype there. And so you have this potential for intercellular interactions through what they call a Bray-Curtis-like metric. And so it's a metric that they're using to characterize the cells and their distance and then their intercellular interactions. So they use a 3D atlas of C. elegans cells. They implement a genetic algorithm to select ligand receptor pairs most informative of the spatial organization of cells. So from the transcriptome data, which we don't talk about a lot in this group, they have a bunch of transcription factors that they measure, and some of those transcription factors represent these ligand receptor pairs. And so then you can actually take the data set and run this algorithm on it to select the ones that are maybe most enriched in certain cells, and that tells you something about like the interactions between the cells. Validating the strategy, the selected ligand receptor pairs are involved in known cell migration and morphogenesis processes. And we confirm a negative correlation between cell-cell distances and interactions. So this means that you have this relationship between cell-cell distance and interactions. So the farther away the cells are, the fewer interactions there are. So they're identifying these interactions and their relationship with intercellular distances, and they can look at this at a molecular scale instead of like a phenotypic scale. So they're trying to link these two levels of explanation. Let me see if there are any images in this paper that are really interesting. Because going through the methods might be a bit much. It's pretty intense. And I'll give you a link to this folder when we're done. But so they're looking at this. So the methods, they have single-cell RNA-seq data. So there's this method called single-cell RNA-seq, which is where they take the cells and they extract RNA, and they're looking at all of the RNA that's expressed across the genome. So there's a sequencing tool that they use to align. What they do is they extract RNA sequences, which are like DNA sequences, but a little bit different. And then they align them with the genome. So you have all these little fragments of sequence that represent what they call transcripts. And they align them with the genome so they can tell what genes are being expressed. And they can do this at the single cell level, which means each cell, you can extract its own RNA-seq profile. And so they have this data set containing 27 cell types at the larval L2 stage, which is, pretty early, moderately early in the larval development. And so they were able to use that as a transcriptome. And then they were able to align this with a 3D digital atlas of the larval L1 stage. So they have this atlas of all the cells, 350 cell nuclei in C. elegans. And they use that for the spatial analysis. So they registered these single cell molecular profiles with cells in this atlas, and they were able to get a sense of, where the cells were in the phenotype, but also what those cells were expressing. And so from that, they could extract Euclidean distances between cell types. So they could build a model, a Euclidean distance between every cell, and then, use these molecular data to say whether those distances are, related to different things that are being expressed or not. And so this is how they get the correlation between molecular activity and cells being either closer or farther away. And so then they also did some bioinformatics here where they looked at different ligand receptor pairs and looked for like C. elegans specific receptor pairs. And there's a lot of detail in here, but basically they're sort of validating the the RNA sequences that they get to say that indeed they are related to these ligand receptor pairs, and that they're strongly predictive of that activity. And then they do this, they use this Bray-Curtis score, which was representing the potential of interaction. So it considers a number of active LR pairs, which are these ligand receptor pairs, that a pair of cells has, while also incorporating the potential that each cell has to communicate independently. So it normalizes the number of active LR pairs used by a pair of cells by the total number of ligands and receptors that each cell expresses. And so they can build, sort of a matrix from that. Let's see. I'm still looking for some figures here. They don't really have any in here that I can... Well, wait a minute. Okay, they're putting them at the end again. Okay, so this is a good example here of what I'm talking about. So you have the CCI score here. You have these cell types. So now you have the cell types, and you have the score, and this basically tells you sort of the cell-cell interactions between different pairs of cells. So you might have like germline cells, which are segregated out from the rest of the cell types, which you should expect. Touch receptor neurons, so you'd have touch receptor neurons across here, other interneurons, and you get this degree of the CCI score, which is the degree of interaction. Body wall muscle versus germline, you can see, or versus touch receptor neurons, you can see it's about the same value. And you have this heat map where you're showing maybe a little bit higher CCI score for cholinergic and pharyngeal neurons, for example, or pharyngeal neurons and ciliated sensory neurons. So neurons are interacting together more strongly. That's maybe what we should expect. And then, so they have that. They have sender and receiver cells. We talked about UMAP a couple weeks ago, this method of doing multidimensional analysis. And so they're using that in this paper to look at sender and receiver cells. And then they're looking at these pairs selected in each run of their genetic algorithm. So they're looking now at the transcripts, and they're looking at the abundance of these transcripts. So transcripts occur at a certain copy number in each cell. So the higher copy number, the more abundant it is, and that tells you something about its activity, that it's higher, it's more active in those cells, which means that if you have two cells that have a ligand receptor that's highly active, that means that they're probably interacting somewhat. So that's the sort of the logic there. But they're using this genetic algorithm to, simulate or resample the data, and then they analyze it in this graph. And then they look at the change here of signaling function for different things, receptor types. So that's kind of an interesting graph because they're using these colored arrows. Yeah, so I think this is a pretty intense paper. I think if you want to read more, I'll give you the link and you can read it. But this is a nice graph here of C. elegans as sort of the phenotype laid out and showing like all these different cell types, their location, and then their degree of interaction here. So that's that paper, and I'll send the link to this. Let me put this in the chat. Okay, Mayukh had to leave, and so did Dick, but I'll go through one more paper, and that's this biological research, and this is something I think Jesse would be interested in specifically. This is Life with Purpose. This is an Aeon article. It's a popular article here, and we talked a couple weeks ago as well about this new paper on sort of non-neuronal cognition. Michael Levin and they wrote this paper on non-neuronal cognition that was called Cognition All the Way Down. Well, this is sort of a follow-up to this. So this is Life with Purpose. And we've talked about this in the past in this group about purpose and biology. The idea here is that biologists balk at any talk of goals or intentions, but a bold new research agenda has put agency back on the table. And so this article goes through, the role of maybe like purpose or goals or intentions in biology. And so some people have suggested that you can use a model of cognition to represent goals and intentions in biological systems that don't have a brain. And so this is a little bit more on this in this direction. So they talk about animal immune systems, for example, which don't have a brain, but they have a sort of a goal-oriented behavior. So they try to knock down invading viruses and invading other things in the body, and they try to minimize the damage to the organism. And so there's this thing called adaptive immunity, which allows the immune system to do its thing, but then you say, well, how can this be if it doesn't have a brain? How can you have this kind of intentional system? So, this description of the immune system, they ask, isn't this an absurdly anthropomorphic way of describing a biological process? Single cells don't have minds of their own, so surely they don't have goals, determination, gusto. When we attribute aims and purposes to these primitive organisms, are we just succumbing to an illusion? And so they bring up this classic psychology experiment, which reveals the human impulse to attribute goals and narratives to what we see. And this is an example of this Heider and Simmel experiment where, so in this experiment, they show people a crudely animated movie featuring a circle and two triangles. And then they ask viewers what's going on in this image. So it's kind of a minimal image of some sort of action, and they ask people what's going on, and they attribute some sort of thing that's, there's some narrative to it. So these triangles and circle and this box, they're escaping this space, and, whereas this could just simply be a phage or a macrophage in biology. So where they could just be abstract geometric shapes, but we assign some sort of meaning to this. And so that's the idea. And so the question is like, if we're observing something at the biological scale, are we just assigning narratives to it when we come up with, say, a theory of how something works or even describing it? Or, are we justified because maybe there is something going on in terms of like, agency or action? And so I think that's a good way to kind of think about this is, to be mindful of, are we just kind of like adding in our own story that, is sort of anthropomorphic and maybe something we would do if these were human agents instead of just shapes or cells or whatever they are. And so, so this paper, it's the author here points out, one of biology's most enduring dilemmas is how it dances around the issue at the core of such a description: agency, the ability of living entities to alter their environment with purpose to suit an agenda. So, in biology, this has been very controversial. People criticize you very much if you talk about like agency, or sometimes if you talk about teleology. But, the question is that really justified, or maybe there is something there? Cells and bacteria aren't really trying to do anything, just as organisms don't evolve in order to achieve anything. That's the idea. And so we want to be careful, but we don't also want to throw the baby out with the bathwater. So, they suggest that a bottom-up theory of agency could help us interpret what we see in life, from cells to societies, as well as in some of our smart machines and technologies. We're starting to wonder whether artificial intelligence systems might themselves develop agency. But how would we know if we can't say what agency entails? Only if we derive complex behaviors from simple first principles, says the physicist Suzanne Still of the University of Hawaii. Can we claim to understand what it takes to be an agent? And so this is then they go through what an agent is. And so, it's a pretty good article if you're interested in that area. I think, there's a lot of discussion to be had about that. I'm not sure what we can apply it to, I think, a lot of problems in this group. But, it's available for people to read. And maybe in future meetings we can talk a little bit more about this. But I know that Jesse and I and some other people have been having conversations about this, which is why I bring it up. And Dick also, I think, has expressed interest in talking about the biology of purpose in the past. I think he's written some things on this, so I'm not really sure. I kind of brought this up after he left, but maybe we'll talk about it in future meetings. So, any comments or questions about those two papers?
Just that I didn't. Thank you for going over that. I didn't get to. That's on my reading list for sure. But I look forward to going over that more in depth and maybe incorporating that into some things here. Yeah, thank you.
Yeah, no problem.
Okay, I'm glad you're going over these things. They're different from what I'm studying, and it's nice to hear about them.
Yeah. Krishna, you're muted if you want to.
Before him. Yeah, okay, go ahead.
Okay. Oh, Krishna, go first, and then Assaf.
I was thinking I have nothing to ask.
Okay.
Yeah.
All right, Assaf.
I love the analogy to the antibody system, the immune system. It's brilliant, and the squares and the shapes, brilliant. But it makes you think. So, do we really think?
Yeah. Yeah, I mean, it's a question.
I love the analogy. I would, because it's so different when you talk to people and you try to tell them it's just tiny robots that make up our brain, and each robot is dumb. So why are we so smart? So now I have a good analogy. It's no different than the immune system. It's perfect. I love it.
Yeah.
Thank you for the paper.
You're welcome. Yeah. Yeah, it's definitely like, even when we talk about theories, it's like we come up with theories and we kind of use analogies, and then are the analogies really appropriate, or are we just kind of making up something that maybe sounds good to the human ear? Like, we think about things like conflict, or we think about things like cooperation. And, there are a lot of theories in biology that rest upon like conflict and cooperation, and at what degree are they really doing things maybe what a human would do, or is it just some cooperative process that's really not reliant on any agency, or maybe there is agency, we don't know, and it's yeah. So okay, well, thanks for attending. If you have any papers to recommend, or if you want to present anything in a meeting, please let me know in advance, and we meet at the same time every week. So next week we have, I think, an open schedule, so if you want to do something, we can do it. Otherwise, I will send out some follow-up to this meeting, and hope to see everyone next week. Have a good week. Thanks.
Bye.
Bye.
Thank you.